Hello, good morning, everyone. We will be doing lecture 2 today, which is blood and lymphatic system. Okay, let's get started. So first question here, list 3 types of granulocytes. Okay, so granulocytes are a type of white blood cells, and what they are basically cells that have visible granules under the microscope. So let's write that down for you. Visible granules under the microscope. Okay, so what are granules? You can think of granules as kind of like those lysosomes that you learned last semester, that they contain chemicals, they contain special enzymes that would help them break down things the cell engulf. And you can see them clearly under the microscope for granulocytes. Okay, so there are 3 types of granulocytes that we talked about in the lecture. Does anybody remember what they are? What are some examples of granulocytes? Thank you, Jonathan. So there is the neutrophil. Okay. There are the eosinophil. And then there is also going to be the basophil. Okay. So what do they do? Well, the neutrophils, they are the most common type of white blood cells. Okay. And they are basically a type of phagocytes. So they go around and eat stuff. Okay, so approximately 70% of WBC are neutrophils, and they are an example of phagocyte. Okay, so phagocytes are cells that go around and they do phagocytosis. So there are different types of phagocytes. One example of phagocyte, it's neutrophil. Okay, so it's possible for a cell to belong to more than one type of categories. In this case, neutrophils can be a granulocyte, and it's also a phagocyte. Okay, it's just a different classification, right? When you say it is a phagocyte, is defining it based on its function, its ability to do phagocytosis. When you are identifying it as a granulocyte, you are classifying it based on its appearance. What does it look like under the microscope, right? That you're able to see their granules. Eosinophil, they, to keep it short, they can fight parasites. Okay, so if you have like a worm or something, they are going to be able to help fight it off. They also play a little bit of a role in things like allergies, but we're just going to keep it simple, parasites. And for basophil is responsible for inflammation, inflammatory response. Okay, so what is inflammation? Well, here are some characteristics which you will learn next class, actually, characteristics of inflammation, right? Just in case you can't really visualize what exactly it means. So inflammation, think about like last time you cut your finger or something. It's going to be red. It's going to be, there's going to be swelling. It's going to be pain. And then there's going to be some heat to it. So all these are characteristics, hallmarks of inflammation. And for the most part, they are brought on by this thing called basophil. Okay, and we will actually learn another cell later on that relates to basophil, and it's also going to play a role in the inflammatory process. Okay, so there you have it, 3 types of granulocytes. Next one, we have the agranulocytes. Agranulocytes, they also have granules, okay? Also have granules, but are not visible. Okay, you cannot see them under the microscope. For some reason, these granules don't really take up the stain as well as the granulocytes do. So you don't really see them, but they're there. Okay, so there are three types of agranulocytes. Okay, three types of agranulocytes. The first one that we talked about in the lecture is the monocyte. Okay, so the monocytes are kind of like, you know, your security guards, okay, patrolling your body and just making sure everything is okay. And if it encounters something that is not supposed to be there, they are going to transform. They are going to differentiate into macrophage. Macrophage. So macrophage means big eater, right? So another example of phagocytes. This is a one-way transformation. After the monocytes become the macrophage, they stay in that form and it continues to fight. Yeah, they fight until the end, until they die. Okay, so only transform if they encounter something that's not supposed to be there. Although in some parts of the body where it is very susceptible to having encounter foreign pathogens, like the lungs, for example, you keep on breathing in stuff. So you would have some what we call like resident macrophage. Okay, they just kind of live there. Okay, it's always in the form of macrophage because your body anticipate pathogen coming in. You breathe them in, they will be ready. Rather than waiting for monocytes to become the macrophage, they are there ready to clear out any invading pathogens. Another two examples of agranulocyte. Anybody? Yep, there are. Very good. That's right. T cells or T lymphocytes as well as B cells. Okay, so T cells, they coordinate immune response, and B cells, they produce antibodies. Okay. So here we go. Let's try this quick question here. Responsible for inflammatory response. So we talked about that already, and that is going to be the basophil. So I'm gonna put B right here. Next one, coordinate immune response. I just told you the answer is T cells. Can differentiate. Differentiate means like specialize. It transforms, right, into phagocytic macrophage. Okay, so you can see the monocyte can differentiate into macrophage. So the answer is E. Attack parasites. That would be our eosinophil. The most abundant phagocytes, the ones that you have the most of, is the neutrophil. Finally, produce antibodies. That would be the B cells. Okay, so antibodies, just to refresh your memory, these are Y-shaped, literally shaped like a Y, Y-shaped proteins that fights pathogens. And pathogens can be like bacteria, virus, and so on and so forth, right? Okay. So if you think about like the coronavirus or something, like the COVID virus that we're talking about, right? They have these spike proteins on them, right? So the spike proteins are kind of like keys, right? And then on our cells, we have receptors. And the receptors are kind of like locks, right? So when the spike protein binds to our cells, they would infect our cells and then they would replicate and cause us to become sick. If you have the antibodies for it, right, because you've got vaccinated, for example, or because you have been previously exposed and recovered, then the antibodies will bind to these spike proteins. Okay, it's kind of like putting gums onto your keys. So the keys will not fit into the locks anymore. And if they can't fit into the lock, they cannot infect. And that's basically how your immune system works. So to make those antibodies, we need to have the B cells. Okay, so exactly how that process occurs, we're going to learn about it next week. Any questions so far? All right, let me see if we have poll question. Let's do a poll question here. Simple one, just to see if you guys are paying attention. Go ahead. Okay, looks like anyone who wanted to answer already answered. And most of you got it right. Basophil, right? And you should know it, right? These are basic factual recollection type of questions, right? You should have no problem answering them. Okay, let's continue with this. Next one, we have list the steps in the coagulation pathway. The coagulation pathway basically means to to stop the bleeding. Right? Coagulation pathway to stop the bleeding. Stop bleeding. Okay, if you have a cut, you need to stop the bleeding. So what happens is the damaged blood vessels will expose the underlying tissue. Okay, remember, if you have a blood vessel, the inside, the innermost layer is going to be lined with smooth endothelium, right? Because blood do not like to pass through a rough surface. So if it's not damaged, then you would have a smooth, uninterrupted endothelium. Okay, endothelium. Okay. And what happens is if you have like a thumbtack coming in or something, then that's going to damage the smooth surface and it's going to expose the underlying tissue. Okay, and the damaged tissue will release something called thromboplastin. Oh, I just realized there is a mistake. All these years, I did not know there was a mistake there. Thromboplastin, T-I-N. Okay, thromboplastin. So that's going to be released from the injured blood vessel. Okay, what does thromboplastin do? Well, circulating in your blood right now, in your plasma, you have a lot of proteins. Okay, and in that plasma, one of the proteins that are related to coagulation is something called prothrombin. Prothrombin. Okay, so prothrombin is actually inactive at the moment. Inactive. Okay, so unless you have some kind of a cut, or unless you have some kind of bleeding going on, the prothrombin will stay inactive. It will stay in this particular form. However, when a vessel is damaged, the thromboplastin that's being released, that will convert the prothrombin into its active form. Okay, and the active form of prothrombin is thrombin. Okay, and in order to do that, you actually need to have a little bit of calcium. Okay, calcium kind of catalyzes reaction. Okay, so prothrombin, once it's formed, once it's activated, will in turn activate another coagulation protein. It is called fibrinogen. Okay, so fibrinogen is also in your plasma and it's inactive. But in the presence of thrombin, it will get activated into fibrin. Okay, so just to refresh your memory a little bit, this is your blood and you put it in a test tube. You can spin it down and separate it. You will have the liquid portion, which is the plasma at the top, and then you will have the cellular portion at the bottom. Okay, so these are like formed elements, okay, which are like your white cells, red cells, and platelets. Okay, so in the plasma, you will have the prothrombin, you will have the fibrinogen. Okay, but when you damage the vessel, the thromboplastin will convert the prothrombin into thrombin, which in turn converts the fibrinogen into fibrin. So you also need a bit of calcium. And what happens is the fibrin is going to form these long strands. Okay, and then they're going to cling to each other and kind of form a mesh. Okay, and this mesh will catch red cells, it will catch like platelets, and eventually if enough platelets sticks to it and red cells stick to it, then you will form like a fibrin clot, which would seal the leak in the damaged blood vessel and stop the bleeding. Okay, and then later on, you will dissolve the clot and replace it with normal tissue. So that is the coagulation pathway. Any question?
Professor, I have a question.
Yes, please.
Does that mean that fibrinogen, to make it active, it's being triggered by thrombin?
Exactly. Yeah.
Okay.
So you need to. It's like one thing leads to another, right? And in order to get thrombin, you need the thromboplastin. Okay. So before surgery, right? They would do something called like PTT, right? Prothrombin test, right? To see how long it takes for your blood to clot. If it takes too long to clot, then they have to transfuse you with these clotting factors, right? So you don't bleed out during the surgery. Okay, number 5. Complete the following chart related to ABO and RH blood group. Just to refresh your memory, blood groups, they are basically the presence or absence of molecules, right? Okay, we call them antigens on RBC. Okay, so we look at your red blood cells, what's on it, what's not on it. Okay, that essentially makes up your blood group. So if you have the A protein, then you're type A. If you have the B protein, then you're type B. If you don't have A or B, then you're type O. Okay, so that's basically what blood group means. Your blood group is determined by your genes. Okay, and that of course is in your DNA. So depending on what allele you have, then you're gonna have different blood types. And all this stuff is explained in details in the lecture recordings. But let's go through this chart to summarize what we learned. Okay, so number of alleles in the population for ABO. How many alleles are there for the ABO blood group in the population? Anybody can help me out with that? Shamira and Safi said 3. That is correct. We have the IA allele, IB allele, and the little I allele. Okay, so 3 in total. Very good. Despite the fact that there are 3 alleles in the human population, each person at most can have how many different alleles? For each person. Okay, so there are 3 out there in the whole population. There's IA, IB, and small I. But for an individual, at most, how many alleles can we have? 2 is correct, right? Because we are diploid, right? Remember, diploid means we have 2 sets of chromosomes. So even though there are so many alleles, it doesn't matter if there are like 4, 5, 6, or 10 alleles out there. We are diploid. We only have 2 sets of chromosomes, so we can have at most 2 different alleles. I hope that makes sense. So what are the possible genetic combinations of these alleles? Okay, so you can have IA, so that would be homozygous. You can have IA small I, right? IB, IB small I, IA, IB, and small I, small I. So a total of 6 possible genotypes. Okay, so IA and IB are dominant, over small I, and IA and IB are codominant with each other. Okay, so that means if you have little I with one of these things, the small I will not show. Okay, and then if you have both IA and IB, then both of them will be expressed. So based on these 6 genotypes, you will have a total of 4 possible phenotypes. Okay, so both IA small I will give you type A blood. Both IB small I will give you type B blood. These will give you AB, and this will give you type O. So total of 4. Is that okay? Please let me know if you have any questions. All right, RH, right? RH, what is the RH? RH is, again, another blood group. There are like at least 30 blood groups on your cells, right? The most common ones are the ABO and the RH. So ABO is whether you have A or B on the cell, and RH is whether you have RH or not have RH protein on your RBCs. Okay, so there are how many alleles for RH in the population? How many alleles for RH, for rhesus? Yeah, Jyoti said 2. That is correct. There is the positive allele and negative allele, 2. And so, how many genotypes are possible with these 2 alleles? Three, yeah. So you can be positive, positive. You can be positive, negative. Or you can be negative, negative. Yeah. So 3 possible genotypes. And then in terms of phenotypes, as in the expressions of the genotype, that would be Rh positive or Rh negative, which would be 2 types. Okay. So that's it. Are we together? All right. Let's do some polls. Yeah, let's do this one. Why not? Just to see if you remember. Okay, so again, most of you got it right. Most of you who participated got it right. It is the thrombin. Okay, thrombin along with some calcium will activate fibrinogen. Okay, so like number 1 and 2, right? They are simple factual recollection questions, right? You just have to look at your notes, or you just have to study and remember the answers, and you will get it. Let's try one that is harder. Okay. Number 3. Okay, just maybe half more minute. I really want you guys to think about this one. If you're not sure, you can choose E. So that way I know you're there and you're participating. All right. So the most popular answer is D, followed by A, and then C. Anybody want to explain why? Anybody want to explain how they got their choice? Just so I know what are some of the thought processes that are going on. No? That's all right. So let's read this again. The MN blood group has 2 alleles, okay? And you don't really care what chromosome is on, but the alleles are LM and LN, okay? If the heterozygous condition is codominant, okay, that means they are both going to be expressed. How many different phenotypic expressions will there be in the MN blood group? Okay, so let's write out all the possible genotypes. How many genotypes will we have? Anybody? How many genotypes for this MN blood group? I think someone says 2. Okay. Let's list them out. So we can have LM, LM. We can have LM, LN. We can have LN, LN. That's all, right? 3 types. Okay. So let's write out the phenotypes now. LM, LM. What kind of blood group would this be? Anybody? Please tell me. What type would this be? No? Type M, right? They're both M. What about LN, LN? What type would this be? That would be type N. What about this one? LM, LN. What type would this be? Anybody? This is MN, right? Because the heterozygous condition is codominant, right? That means they are both going to be expressed. So therefore, 1, 2, 3, 3 different types. I know some of you are probably a little bit confused, but I don't know what you're confused about if you don't let me know. Okay, so ask me something so I can address your concern. Right? Otherwise, if you get this question on the test and I change a few wordings, you might not be able to get it. So any questions? No? Everybody's happy? All right. All right. Let me change something. The if the heterozygous condition, okay, so instead of saying that, I'll say if LM is dominant over LN. Okay, how many phenotypic expressions will there be? Let's try this question again with that change. Let's see if you guys will get it this time. of the 5 people who participated, you all got it right. So this would be type M. This would be type N. And because M is dominant over N, when you have them together, it is still going to be N. All right, so there you have it. A harder question that's worth thinking about. Okay, let's move on. This chart was filled in the lecture recording, so I'm not going to do it again. Let's move on to number 7. Okay. Complete the following chart related to ABO and RH by group. This is talking about blood transfusion. Okay, so A plus. If you're A plus, who can you receive from and who can you donate to? All right. So there are when you look at whether a transfusion is safe or not, you have to ask 3 questions, right? So safe transfusion. Okay, so you have to look at 3 questions. Number 1, okay, antigens on donor's red blood cell. Okay, what do they have? Do they have A? Do they have B? You know, do they have nothing? Do they have both A, B? What kind of antigens do they have? Number 2, antibodies in recipient's plasma. Okay, what kind of antibodies do they have? Okay, and number 3, will the antibody cross-react with the antigens? Okay. So let's just do a very simple question here. Let's say I take A blood and give it to an AB person. Okay. This is the donor. This is the recipient. Is this safe or not safe? Okay. So if you're not sure, you can use these 3 questions to think about it. First one, antigens on a donor. Okay. So if you're a type A person, then on the RBC, all your red cells, you will have type A antigen. Okay, so I can draw it out. There is going to be the red cell, and you will have A antigens all over the place. Okay, let's just show 2. The antibodies of the recipient's plasma. Okay, so you're going to give it to an AB person. Remember, you make antibodies for anything that you do not have in your body. Okay, so if you're AB person, you will not be making antibody that would attack A or B, because otherwise you will end up attacking yourself. So in this case, you will have no antibodies against A or B. So in this case, because the recipient doesn't have any antibodies, it's okay. You can give this to them and there will be no reaction. Therefore, this is going to be safe. Okay, let's try another one. What if I give B blood to an A person? Is this safe or not safe? Okay, you know what? Let's do AB blood to an A person. Is this safe or not safe? Right? So first question, antigens that are on the donor's RBC. If you are AB person, you will have A as well as B. And then you're going to transfuse it to a type A person. And if you have type A blood, then you're going to make antibody that would attack something that you don't have, right? And you don't have B, so you're going to make anti-B antibodies. So when you go ahead with the transfusion, these anti-B antibodies will attach to anything that has a B on it. So they will attack and destroy all the incoming cells. Therefore, this is not safe. Does that make sense? So to have a cheat sheet or help sheet, whatever you want to call it, on the test to help you get this stuff sorted out quickly, you can use this chart, right? So let's say O, right? And then AB and then AB like this. Okay, so this is a transfusion flow chart. It tells you who you can give it to. So O can be given to A, and A can be given to AB. O can be given to AB as well as B. And then B can be given to AB. Okay, so O is sometimes called the universal donor, okay, because you can give it to all the other blood group, right? But like I said in the lecture, it's not entirely true because while O is compatible within the ABO blood group, there are so many other blood groups, right, that you have to consider. You can't just automatically assume someone who has type O blood can give it to everybody. Like, what if they are Rh positive? You cannot then give it to someone who's negative, right? Okay. There's a question. Okay, a question not related to what we're doing, but it says, is there a protract or lockdown browser for the test? No, none of those stuff. Okay, it's just a regular test. You don't have to install anything. You just do it as is. Okay, and it's okay to use your notes. Okay, I'm gonna throw it out and say it there. Okay, some people don't use their notes, which is honorable, and I respect that. But there's no sense of not using your notes. Okay, but don't flip through your notes, right? Like, you won't have time. Write down everything on like a summary sheet, and then you can have quick reference. Okay, so something like this that I'm writing right now, you could have it in your help sheet. Okay, it's okay. It's not cheating. In fact, it's probably going to be necessary. Okay. Next one is RH. For RH, it is going to be negative to positive. Okay, negative people can give to positive, but not the other way around. Okay, you will sensitize them, and that's not a good thing. Okay, so having said that, please tell me, somebody please tell me, A plus. If you're A positive, who can you receive blood from?
A positive and A negative.
Yes, you can receive from another A positive. You can receive from another A negative. Very good. Anyone else? Who else can I receive it from? Yes, we can also receive from O positive and O negative, right? Take a look here, right? If you're A, you can, of course, receive from your own blood type group, but you can also be receiving it from type O. Okay? Type O is good, right? O has no A or no B on it, so you can give it to other blood groups. Okay, who can you donate to? If you're A positive, you can donate to, of course, other A positive. And you can also donate to AB positive. Okay. Can I donate to anyone else? Can I donate to AB negative, for example? No. Yes, a capital no from Shamira. That's right. Okay. If you're positive, you cannot give it to negative people. Okay. Very good. So B is similar, so I'm just gonna do it quickly. Okay. There we go. Okay, let's take a look at AB positive. Who can you receive it from? AB positive. Okay. AB positive, you can receive it from, basically everybody. O positive, O negative, A positive, A negative, AB positive, AB negative. B positive, B negative. Okay, so if you're AB positive, you've hit the jackpot. You can receive blood, RBC, red cells from everybody. I'm AB positive, I hit the jackpot. Who can you donate it to? Just other AB positive. Okay, no one else. Is that clear? Now, just to emphasize, because I don't want you to get the wrong idea, we are talking about RBC transfusion. Okay, we do not transfuse whole blood. Okay, we take blood. If you donate blood, they would take the blood and they would separate the plasma from the RBC. Okay, the white cells we cannot donate because the white cells, they would attack everything, right? Okay, and we're talking about just taking the red cells and giving it to other people. The plasma transfusion, that follows a completely different set of rules. Okay, so that's not what we are talking about. We are only talking about RBC. All right. Okay, so very quickly, if you're O positive, you can receive from O positive and O negative, and you can give it to A positive, B positive, O positive, and AB positive. All right. I will do one of these negative stuff, and then you can fill in the rest yourself. Okay, if you're A negative, who can you receive from? Well, you can receive from A negative and O negative, no one else. And you can give it to, excuse me, A positive, A negative, AB positive, and AB negative. So I hope that is okay. If you have a question, please do let me know. Otherwise, we will take a moment and fill this in. Okay, go ahead. Sorry, guys, I forgot to fill in the screening form for my kids for school. I'm just gonna do that quickly while you guys work on this. Okay, so find out which one is safe, which one is not safe. You're taking these blood and then you're giving it to the recipient shown here. So you have to tell me whether it is safe or not safe. Okay, well done. I am going to write the answer and you can compare it to yours. Okay. I think those are the only safe ones. Does anybody want me to explain a particular one that you're not sure why it is safe or not safe? I can explain. Anybody? O negative and A plus, like this one, like why is it safe? Okay, let's see. So O negative to A plus. Okay, 3 questions, right? First question, what is on the donor's RBC? Nothing, okay? If it's type O, there's no A, there's no B, right? O is the absence of A and B. Okay, there's no O antigen, okay? O is absence of A and B, okay? And negative means there's no RH either. So there's nothing as far as ABO and RH is concerned. And then you're going to give it to someone who's A positive. Okay, so if someone is A, they would have anti-B, right? And that's okay because there's nothing on the incoming cell. The antibodies will not recognize it, right? And there's no cross reaction, therefore safe. Okay. There's a private message. Why is all the A plus not safe? Okay, remember positive cannot be given to negative, right? Okay. If there is positive, then there's RH on the cell, and then negative people, they would make anti-positive antibodies, right? And they would attack it. So despite the fact that A can be given to AB, but positive cannot be given to negative. That's why it's not safe. Okay. Which page is this information to fill out? I am not sure. I don't have the physical workbook, and I don't quite remember. Maybe this one is not in the. I think it is though. I think it's in the workbook. But if it's not, then please take a picture and then you can fill it out afterwards as well. Okay. Any other question before we continue? Okay, let's do another poll question right here. This is a multi-select question. There you go. Person with A negative blood can receive RBC transfusion from, and try not to look at the chart, but if you must, then you could. Can I scroll scroll down like this? Sorry, which way? That? Oh, okay. We're doing number 4. Okay, I am going to end it soon. Can you please show the chart with all different blood types? Okay, we'll do that in a second, okay? Let's finish this. All right, end the poll, share the results. If you're A negative, who can you receive from? All right, so again, right? Use this chart. If you cannot remember, okay, you can use this full chart. If you're A, you can receive from other A's and other O's. All right, so definitely not AB, definitely not B. You can get it from other A's or other O's. And then this is a negative person. Negative can only receive from other negatives, so not positive. The answer is A and D. All right. Okay. Which chart are you talking about, Patrick? Are you talking about like this one or like this one? This one? Okay. Do you need to take a screenshot or something? You know what? I'll post this afterwards anyways, right? So you guys should be okay. All right, moving on. What is this stuff called? Pedigrees. So Josh's father has type O blood. If Josh has type A blood and he marries Kat with AB blood, find the expected blood type for their kids. Okay. So we can do a family tree if you want, but we don't really need to. We can just, think about this. So Josh's father is type O blood, right? So what is the genotype rather? What is the genotype for someone with type O blood? Yeah, little I, right? For certainty, we know it is little I. Josh has type A blood, okay? So type A blood is a little bit tricky. Like, is it IAIA or is it IA little I, right? We normally cannot tell unless we know their family history. And in this case, yeah, we do know, right? Because the father is type O, right? So the only thing the father can pass on is a small I. So Josh must have got the A from his mother, and he has to inherit the small I from the father, right? It's guaranteed. And then he marries Kat. Okay, if someone's AB, we know their genotype has to be IAIB. So when they get married and have kids, right, half of Josh's sperm will be carrying the IA allele, the other half will be carrying the small I, right? During meiosis, right, the chromosome separates. And for Kat, half of her eggs will carry IA, the other half will carry IB, like so. Right? So if this sperm finds this egg and they unite and fertilizes, then you will get IA. This and this can come together to form IA small I, and so on and so forth. So if you do the calculation, there is a 50% chance that their kids will be type A, 25% chance type AB, and another 25% chance of type B. That is how you do it. Okay. Now, of course, that is the easy one. Let's do a harder one. Okay. Right here. List all possible blood types for question mark right here. Has anybody done this yet? If you have done it, feel free to share the answer. If you have not done it yet, I'm going to give you maybe like 2 minutes to give this a try, and we will take it up. And again, right, like I don't mean to be a nag, but you really have to watch the recordings before coming to the lectures. I'm looking at the online stats. Less than half of the class have watched the recordings. Okay, this is not a substitute for the actual lecture. You won't do very well if you just come to this and not do your own reading, if you don't watch the lectures on your own time. Okay, so Safi said IA small I is what she got. Okay. Anyone else? If you don't feel like typing your answers in the public chat, you can private message me. That works as well. Okay, that was 2 minutes. Last call. Anyone else want to chime in? Tell me what they think the answer is before I take it up with you. All right. So to do a question like this, we will write down the blood type of everyone that we know first. Okay, so Megan agrees with Safi. Very good. Let's see. So here, type O is little I. That's for certain. And for everyone else, we know at least half of their genetic makeup. Okay, that's an easy part. Then you got to think about it. You got to like use logic, try to, deduce the answer. So let's take a look at this lady here. She is type O, and we know that one of the small I came from her father, and then the other small I came from the mother. Okay, so this small I probably came from mother, and then this small I probably came from the father. Right? So that's how we figure out that they are heterozygous, type A and type B. All right. What about this person here? Okay, what about this person? Well, if she is IA, where did she get the I? Did she get the I from the father or from the unknown mother? The IA, did it come from the dad or the mom? What do you all think? Mom or dad? Do we know? Has to be from mom, right? Has to be from mom. Because if it came from dad, then he would have been like type AB. Okay. In fact, we know based on this information, we know for a fact that the father must be IB small I, and that he has passed on the small I to the daughter. Okay. The father could not have been IB, because if that's the case, then the daughter would have at least one copy of IB, which she does not. So we know for certain there's a little I involved here. Okay, so that is as much as we can come up with so far. I mean, we do know this IA came from the mother, and then this IA in turn came from the grandfather, right? Okay. So now the question is, what is the other genotype? Right? Like, what is the other allele? Okay. Because the IA came from the father, her father, the question mark must come from the mother. The question we have to ask ourselves is this: do we know for certain, right? Do we know which of these allele get passed on to question mark? Do we know if the IB got passed on or the small I got passed on? Do we know for certain which one get passed on? Okay. We don't. We have no idea which one got passed on, right? This can be IB or small I. We don't know, right? Because they only have one kid, right? And that kid inherited the IA, right? There's no information about this other one, right? So it could be IB or small I. So the possible blood type could be AB or type A. Okay. What about this? What if they have another kid? What if they have another kid? Let's say they have a son this time, and then this is a type B son. Will we know for certain then? Okay, if they have another son, if they have another kid and it's type B, will we be able to tell whether question mark is AB or type A now? Safi say yes. Which one would it be? If they have a type B son, do you think it's going to be type AB, or do you think it's going to be type AB blood or type A blood for the mother? Think it's going to be I? Okay. Anyone else have a different opinion? Do you guys understand my question? I'm saying if they have another kid that turns out to have type B blood, will we be able to rule out one of these blood type for the unknown? Okay. The answer is no, we still don't know. Okay. So type B could be IB or IB small I. Okay. Let's say it's IB, right? Then like we don't know which one it is, right? So like this kid could have gotten the IB from the father, and then they could have gotten the IB from the mother if she was AB. Okay, but it's also possible that the son here got the IB from the father and then got the small I from the mother if the mother is type A. Remember, if the mother is type A, then it's IA small I. This is IB. So even if they have another son that's type B, we still don't know. Okay, there is still no way to tell. So my next question is, what kind of kid do they have to have in order for us to definitively rule out one over the other? Okay, let's say they have a third kid. Third kid. Yeah, let's have another daughter. Why not? What kind of blood type would this have to be in order for us to definitively rule out one over the other? We only have two blood types left, right? A, B, or O, right? Which one would it be? Exactly, type O. Right? If they have a type O kid, then we know for sure one small I came from father, the other small I must have came from mother, right? Okay. Otherwise, we won't know. We will never know. There's another question in the chat. How are we able to tell for type B, but not for type A? Okay, so again, right, for type B, we know it is IB small I, because over here, the A must have came from the mother. The A could not have came from the father, because if it did came from the father would have been AB blood and not B blood, right? That's the first thing to understand. So second thing to understand is that the father being type B is either going to be IB or IB small I. And then the reasoning is this: he could not have been IB, because if he was IB, he would have passed on the IB to the daughter, making her blood type AB. Right? The fact that her blood type is A tells us the father has to be IB small I. Are we okay? It's not going to get harder than this, okay? And at most, you're going to have like one or two of these questions, all right? So like, don't lose sleep over it. Do try to understand it though, okay? Because it's you know, it's something that you should learn, but you know, don't get too put off by it. Any follow-up questions before I move on here? Everybody's happy? All right. Let's do a few, you know what, let's take a break first. Let's do a 5-minute break, and then we'll do some poll questions when we come back. Okay. If you don't want to break, then you can try this question while you wait. Okay. 5 minutes. Okay. I'll see you at 9:40. All right. Anybody get the answer for this one? We launch the poll and see. Okay. If you haven't had a chance to do that yet, that's okay. I'll take it up right now. But for anyone else who tried it, you can put in the answer. Okay, so as you're doing that, I'm going to explain. So here we have to find out the genotype for John. Where's John? John is right here. All right. So Sue is little I. Mary is IAIB. And then we got a bunch of other people which we might or might not need to know. So let's work with this for now. Harriet is IA something. Now it's always good to look at the relatives of to look at the parents of someone who's type O. Okay, so if Sue is type O, we know one of the small I came from Harriet and one of the small I came from John. Okay, so this one from mother and this one from father. Okay. And let's take a look at Mary here. Mary is IAIB, so the IA must have came from Harriet. And that means the IB must have came from John. Right? So the answer is B, right? Which is what most of you got, the people who participated. Does that make sense? Any question? People who chose D, do you need me to explain? Do you have any question about why it is not D. All right, let's try it one more time. Same family. Who is what is Mona? What's the blood type of Mona? All right, while you're doing that, I will take it up. So same story. Okay, the choices are different though. So let's take a look here. Mary is IAIB. So we know Harriet is IA small I, John is IB small I, just like before. Okay. So what about Ed? Ed is small I, right? So Harriet got the small I from Ed, which means the IA must have came from Mona. Okay. But what's the other part? What's the other part? Do we know? Well, they only have one child, only have one daughter. So we don't really know. The other one, for all we know, it could be IAIA, it could be IBIB, could be IAIB, could be IA small I, right? There is really no way for us to tell. So the correct answer is need more information. Okay, it's not either. You know what? None of these make sense. So the answer is need more information. It could be IAIB, IA small I, or IAIA. We don't know. Okay, maybe we need to find out what her parents are, so on and so forth. Is that okay? I certainly hope so. All right. Now that I have everybody's attention, sort of, just want to go through the quiz next week, right? So there's some, sorry, where is this? There's a quiz next week, your first quiz, and the quiz information is posted on eCentennial. It's going to be available on Tuesday from 6 to midnight, essentially. 40 minutes time limit. And there is the quiz link. You can click on it. I'll create another hyperlink for next week's to-do list. You can just click on that as well. There will be 15 multiple choice questions, 4 application questions. So application would be like the harder ones, right? Like the LMLN thing that we did, that would be like an example of an application questions. 4 multi-select questions. And the total is going to be 27. Okay, 27 marks. And if you need accommodation, please do contact me, and I will make sure you get extra time and stuff like that. All right. You do also have to watch the recording next week. Right? Just because there's a quiz doesn't mean there's no lecture. Next week we'll be doing, lecture 3. Any question about the quiz? Of course, there's a practice quiz here that you can do. I will also post like Kahoot link for you to try as well in this week's debrief. And yeah, let's continue then. Oh, there's another question here. Number 5. A person with O negative blood marries someone who's AB negative. Which of the following is a possible blood type for their child? Let me relaunch the poll. Try that one more time. You should not be doing a Punnett square. That's not how you do these type of questions. All right. Oh yeah, there are two AB negative. Oh, thank you for letting me know. Let's change it to positive then. Okay. Sorry. Oh, you know what? Nobody chose a D. I'm going to change that one to positive then. All right. So to do these type of questions, you have to remember four things, right? Okay. Number one, O person cannot have AB kids ever. Okay. Doesn't matter who they marry, because if you're a type O, you can only pass on a small I, and there's no small I in AB. Same thing. AB person cannot have all kids. Okay. In order to have all kids, you need to pass on a small I. If you're AB, you don't have a small I. Number three, O plus an O will always give you O. If you're type O, your spouse is type O, whole family is going to be type O, because that's all you guys can pass on, right? Little I and little I. And the fourth one, negative, and then other negative, whole family is going to be negative for the same reason. Okay, so these are the things that you have to remember when you do these type of questions. Okay? Definitely put it on your help sheet. There's a question. I don't think I will go. Yeah, sure. Okay. Like if you still don't understand it after you watch the lecture recordings and then, you know, go through the tutorial again, you don't get it, just, you know, send me an email. We can set up like some kind of help session or something. Okay. Yeah. All right. So what's going on here, right? The person is O negative, and then the other person is AB negative, right? So process of elimination. Type O people cannot have AB kids. So that's not the answer. That's not the answer. Okay. Type AB individuals cannot have O kids. So that's not the answer. They're both negatives. Negative people can only have negative kids. So that's not the answer. So the correct answer is C. Do you guys understand like these statements? Anyone want me to explain a particular one? You understand how I get this answer, like without much effort, right? Because of these statements. Again, these will be important because like even with the matching stuff, you're gonna need this. Okay, so let me explain one more time. Okay, first statement. O person, a person with type O blood, that's what I mean, right? A person with type O blood, doesn't matter who they marry, they will never have AB kids. Okay? If you are type O, if you're type O, what is the genotype for type O? Okay. Mary, do you know the genotype for someone who's type O? Or Patrick, do you know the genotype of someone who's type O? Not to put you on the spot, but just, you know, checking. Yeah, exactly, right? Small I, small I. Okay. If you are small I, then you are guaranteed to pass on a small I to the next generation, right? Doesn't matter who you marry, you are going to be passing on a small I, which is why you will not be able to have an AB child, right? Someone who's AB, it's IAIB. You don't have an A to pass on, you don't have a B to pass on. So if you type O, you will never have a type AB kid, not a biological one anyway. Does that make more sense now, I hope? Now, similarly, if you are AB, type AB, right, you can only pass on A or B, which means it's not possible for you to have an O kid because O kid needs to get one I, small I, from each parent, right? You can't provide that small I if you're AB. For that reason, if you're AB, you won't be having O kids. If both parents are type O, then they are little I and little I, right? And that's all they're gonna have. Their whole family is gonna be little I, right? Same thing for negatives. If both are negative, then they can only pass on negatives, okay? So based on these statements, again, see, one of the person is type O, right? So automatically I cross out all the A, B answer. It's not possible for a type O person to have an A, B kid, right? The other person is A, B, so I cross out everybody who's type O. Okay. They're both negative, so not this guy. That's why the answer is B minus. I hope that helps a little bit. And if not, we can always email me or stick around after class, and we'll talk more about it. Okay, here, next one. 4 babies are born, and they mix up the babies. Okay, so we're gonna find out who's the father and who's the mother. Now, question 11 I already did in the lecture recordings, so I'm not gonna do that again, but we'll do 12 together. And we'll do 12 together. Okay. And by the way, we don't do this in real life, right? No one's gonna figure if babies got mixed up, no one's gonna do a blood type to find out whose babies are who. We do DNA fingerprinting, right? This is not an accurate way of figuring out. It only works in certain conditions. Okay. So these questions, of course, are set up so that they do work out, but in real life, we don't really do that. All right, so let's see. Here are the parents and here are the babies. Okay, let's find out what the babies are. Let's take a look at the first couple. Okay. A, B, negative for mother, O, negative for father. So using the rules that I've just written down for you, if the mother is A, B, okay, please tell me which of these babies are definitely not hers. Okay. If the mother is AB, which baby is definitely not hers?
Baby E.
Baby who? Sorry, I just didn't hear that.
E.
Based on the mother alone. Baby F, that's right. Yeah, baby F is not hers, right? It's not hers because AB cannot have O, right? Okay. And then the father is O negative. Definitely not that one, right? So 2 babies are out of the pictures. Okay. Then we will check the other 2. Okay. One is A positive, one is B negative. Which one is theirs based on their blood type? Both the mother and the father are negative. So rule number 4 can only have negatives, right? As such, this is not their baby. And they are taking home baby H, baby Hillary. Okay, so that's gone. Do you understand? You don't have to do any Punnett square. You just use the logic based on those 4, whatever you want to call them, 4 statements, 4 things that I told you. Okay, so baby H is gone. Let's take a look at the next couple. A positive and O positive. Who are we taking home? Not the first one, because O cannot have AB. Okay. And what about this one? Which one are we taking? Do we know which one is theirs based on the information we have so far? Not really, right? It could be F, it could be G. We don't know yet. Okay. So we're gonna have to keep on going to see if we can eliminate more candidates. Okay, so next one, O negative and B positive. Right, so if you're O, you are not going to have AB. So is it O positive or A positive? Well, none of the parents have A in this case. So if neither of them has an A, then it's impossible for them to have a type A baby. So G is definitely not theirs, which means F will be their baby. Okay, and since F is the only possible baby for the third couple, we can cross out F for the previous one. Okay, and so for the last one is going to be E, and that is the answer. Okay. So I encourage you to go through this again yourself when you have a moment. Think about it. Let's see. O can't have O baby. O can have O baby. O cannot have AB babies. Okay. O, it's okay for a type O to have a type O baby. For example, let's say a O marries a B, they could still have an O baby. And this is one of the things that people get confused, right? Like, how is it possible? Where did the B go, right? Well, O is little I, and B could be IB, or IB small I. So if he is or she is heterozygous, then it's possible for this couple to have a type O.
Can you explain how two positives can make a negative?
Absolutely, for the same reason. Okay, if two people are Rh positive, right? We don't know if they're positive or positive negative. If they happen to be heterozygous, right, and you do the Punnett square, right? Let's take a look here. Half of it would be positive, half of it would be negative. Same thing for the other parent. And then these two will make positive, negative, and negative. So if they are both heterozygous positive, then there's a 25% chance that their kid is going to be negative. Okay, do you understand? Right. So sometimes it's like hidden, right? You know what I mean? It's the same thing as like two people with brown eyes. How can they have a child with blue eyes, right? Well, maybe they have a small b, like a big B, small b times a big B, small b, right? Okay. All right. I know this is a little bit heavy and confusing, but I promise you it should get better after you sit on it a little bit. Okay. This is not on the quiz, right? I want to emphasize this is lecture 2. Okay, the quiz next Tuesday is only on lecture 1. This will be, however, on the test, and we will have a chance to review before the actual test. Okay. Okay. Yeah. What else are we gonna do? Sorry, I'm kind of getting a little bit confused here. Okay, let's do another poll question. Because why not, right? Relaunch. Okay, there we go. Go ahead. Okay, if the word glycoprotein confuses you, we can just call them proteins. Okay, glyco just means sugar. Okay, sugary proteins. Right, I think everybody who wanted to answer already did. And the correct answer is B. Okay, there are anti-O antibody in the plasma. There's no such thing as anti-O antibodies. Again, O is the absence of A and B. If you don't have A or B on the red blood cells, then you are type O. You cannot make an antibody that attacks nothing, right? Okay, so anti-O antibody does not exist. Someone who's AB blood will have no antibodies against A or B. That's it. Okay. So some people choose C. Let's see why C is not the answer. It is safe for this person to receive RBC transfusion from a type O donor. Right? If you are AB, right, you can receive from O. You can also receive from B, and you can also receive from A, right? So there is nothing wrong with this statement. We're looking for the false one. We're looking for the one that is incorrect. Okay, and so B is the incorrect statement. All right, one more of these confusing who can have what kind of kid question. All right, go ahead. Number 9, we're doing number 9, okay? Okay, I only have 4 people who've answered. Just give it another half a minute or so. Okay. Let's take a look. We're looking for the true statement, the one that is correct. So first one, a person with type O blood will not be able to have a biological child with type A blood. Nonsense. Of course they can. If they marry someone who's type A, for example, or AB, then it's possible for them to have an AB child. So this is not correct. Next one. And RH, well, if this is not correct, then this one is not correct either, right? Okay. So it's only between B and C. An RH positive person who marries another positive person will only have biological children who are also positive. And again, right, this on face value looks correct, right? Positive, positive, of course they're going to have positive. But I just explained earlier, right? You don't know if they are homozygous positive or heterozygous positive. If it happens that this is like heterozygous, if this person is like plus negative and also plus negative here, then there is a 25% chance I showed you earlier that they will have a negative child, right? So it's not certain that they will only have positive kids. For that reason, this is not true. Next one. It is possible for 2 people with type A blood to have a biological kid to type O. Right? That is correct. Right? That is correct because, again, type A could be A little I, right? So if it's that case, then 25% chance, small I, small I. Okay, so you got to be careful with these questions. It's really messes with your mind a little bit, and you have to be careful. Sometimes you might need to read it, a couple times before you make a final decision. Any question about anything we've talked about so far before we go back to our study guide? All right, good stuff. So here, this is, the experiment that we would have done if we are, doing this in person. You're gonna have some antibodies, and you're gonna add like simulated blood to it, and you're gonna figure out the blood type. Okay, so if you take the blood sample and you mix it with anti-A antibodies, and you can see all these little flaky stuff, right? That is a sign of agglutination. Agglutination. Agglutination is just a fancy word for clumping. Okay, so clumping occurs. Okay, this happens when the antibodies react, right? Cross-react. We say cross-react with the antigens. Okay, and it will result in hemolysis, right? The breaking of red cells. Okay, if you have massive hemolysis, it's a very serious thing. You know, your red cells is going to be ruptured. You're not going to be able to circulate oxygen. It could potentially be fatal. Okay, so we don't want that. Okay, first one. Your sample reacted with anti-A, but not B. Right? What does that tell you? Well, that tells you that your sample has A antigens on it. Okay, that's the antibody will only react with the sample if your sample has that antigen, right? Okay. It also reacted with RH, which means the blood has RH. So we call that positive. So first one is A positive. Next one, sample B. No reaction with antibody A. That means there's no A. Yes reaction with anti-B, and yes reaction with RH. So this must be B positive. Very good. No reaction with A, no reaction with B. So if you don't have A, don't have B, you must be O and positive. This is complete opposite. Yes reactions with A and B, but no reaction with RH. Then the person is AB negative. Okay. Anyone else? I mean, any other question? No questions. Okay. So more here. I know it's not very clear in the book if you got the book, but let's zoom in here. You can see all these. That's what it looks like if we were to do it in the lab. This is the kit that I got, and then you see all these fuzzy stuff. That's agglutination. Okay, so this is reacting with the B only, so that's B negative. Okay, so got a private message. How can you tell what the blood type is? Okay, so again, right, you want to see whether or not there is a reaction. If a reaction is observed, if there is a reaction with the antibody, then you have that antigen. Okay, so yes reaction with A, right? So you write down A. Yes reaction with B, so you write down B. Yes reaction with RH, so we write down positive. If there's no reaction, you do not write down that letter. That's basically what it means. Okay, so here, reaction with A, yes, so we put down A. Reaction with B, we put down B. No reaction with RH, we put down negative. And then finally, O positive. Okay. Let's do the fill in the blanks. I think I'm gonna post the answer for the fill in the blanks afterwards, and you can just check yourself. I rather spend time going through some of these long answer questions. Anybody have any requests for the long answer questions? Give you a moment to read them through. You should really read them through beforehand and then you're ready to ask them during the tutorial. But I'll give you a moment to look at them. Number 6, yeah, that's a common one. There's another question. So if the blood sample reacts with A, does that mean the sample is A? It means it has A. It cannot guarantee it's A. You also have to look at whether it reacts with B, right? If it also reacts with B, then it's AB. If it only reacts with A but not B, then it would be A. Okay. Any other requests? All right, so we'll do those first. Let's start with number 6. Okay, so number 6. How does sensitization result in? So sensitization, and then hemolytic disease of the newborn. So again, this is explained in a lot of details in the recordings, but I'm just going to give you like the condensed version here. Okay, so let's talk about what the meaning. What does it mean by sensitization? Okay, sensitization. So we're gonna talk about this in the context of RH. Okay, so an RH negative person can get sensitized if they are exposed to RH positive blood. Okay, that's the first thing to know. Okay. Then what? Okay, what does it mean by to get sensitized? Once sensitized, the person's immune system will make anti-RH antibodies. Okay, so I'll give you a moment to copy that down if you wish, and then I will explain a little bit more. All right. So help me out here, okay? So that I know you understand. Antibodies, right? We make antibodies to attack things that we don't have. We make antibodies to attack foreign antigens, okay? Antigens that are from outside of the body, right? Okay. So for example, you know, if you're type A blood, what kind of antibodies are we gonna make? Yeah, you're gonna have anti-B antibodies, right? Okay. So for the purpose of what we're talking about, when you are born, you will automatically have these anti-B antibodies. Okay, and if you are type B, you would automatically have anti-A antibodies. However, for most of the foreign antigens that you will encounter in your life, you won't start to make the antibodies until after you have been exposed to them, right? Okay, case in point, you are not gonna have the antibodies against the COVID virus until you either get COVID naturally or you get vaccinated, right? Yeah, you won't make those antibodies until your immune system has been introduced to this foreign antigen. Right? You're not gonna make anti-chickenpox antibodies until you have contracted chickenpox or you get vaccinated for it, right? Okay, so RH, okay, is the same thing. Even though RH is a blood group, unlike the ABO, where you are born with the antibodies, for RH, you will not make those antibodies until you are exposed to it. Okay, so who makes RH antibodies? The positive people or the negative people? Who has the potential to make the RH antibodies? People who are positive or people who are negative? Only RH negative people can be sensitized, right? Only they can make the antibodies. If you are positive and you make anti-RH antibodies, you will end up attacking yourselves, right? And that's not a good thing. Okay. So here's what happens. If you're a negative person, you are not automatically born with anti-RH antibodies. How do you get them? You will get them after you've been exposed to positive blood. Okay. And when that happens, we say you are sensitized. Okay, so in the lecture recording, I said normally positive blood cannot be given to a negative person, right? Okay, but technically speaking, everybody gets one free pass. Okay, because if you have not been previously exposed to positive blood, I can give you the positive blood. You don't yet have the antibodies, and so there will be no cross reaction. But once you've been introduced to positive blood, you are now sensitized, and your immune system will start to make anti-RH antibodies. So this is the context, okay, of what sensitization means. Okay, so how does a person get sensitized? How can one? Okay, two reasons for sensitization. Okay, like why would a negative person be exposed to positive blood? Why? Well, one reason is blood transfusion, right? Okay. Maybe someone made a mistake. Someone didn't have their morning coffee and then they got all tired and gave the wrong blood. Could happen, right? Not gonna kill them the first time, but it will sensitize them. Or we ran out of negative blood, right? And this person, if we don't give them blood like within the hour, they're gonna die for whatever reason. They don't have enough blood. We have to save them, so we will give them the positive blood. Okay, we know the person has not been previously sensitized. We know it is going to be safe to do it just once, so we will do it and save it, and then the person becomes sensitized. So that's one reason of how a person can get sensitized. What is another reason why someone might get sensitized? Anybody? Anybody who? Yeah, the Patrick said the placenta breaks and then the child has positive. Exactly, right? So pregnancy. Exactly. Okay, so if you have a RH positive fetus in an RH negative mother. Okay, so long story short, what happens is, let's say the first pregnancy, again, to get the full explanation, you got to watch the thing. Okay, the recordings. So first pregnancy, let's say the mother is negative, and so her blood will not have any RH on them. And then the baby is positive, so there is RH on it. And the baby is positive because of the father, right? Okay. So separating the mother's blood and the baby's blood is the placenta. Okay, so if the placenta is damaged during pregnancy, or a high chance of getting damaged during labor, what happens is some of the baby's blood will go into the mother's bloodstream. So now the mother system is sensitized. Okay, it takes about like 2 weeks to make the antibodies, right? 1 to 2 weeks. So by the time the antibodies is created, the baby will be delivered. Okay, so no problem for the first baby. But in the second pregnancy, right? Second pregnancy, same mother. But this time, her bloodstream would have the anti-RH antibody, right? Because she was sensitized from the first pregnancy. Okay, and so the baby, if the baby is negative again, if the baby is positive again, then some of these antibodies will cross the placenta, and then it will attack the baby's blood. Okay, so the baby will have hemolysis, okay, and lots of hemolysis in the baby. So the liver and the spleen will be enlarged because the spleen and liver is what deals with the broken RBC, right? They're gonna have jaundice because the hemoglobin is going to be broken down into bilirubin, right? They will have edema, right? Without the red cells in their system, you don't have as much osmotic pressure, and all the fluid's gonna leak down, right? So all these things it could result in hemolytic disease of the newborn. Okay, and the severity varies. Okay, sometimes it's very severe, sometimes not so much. Okay, and it's not restricted to Rh. It could happen to any of the blood group. Okay, it could even happen in the ABO blood group. I think I mentioned this in the recording. I'm AB, and my wife is O, and both of my daughters are type B. Okay, so because my wife is type O, she has anti-B antibodies, right? So the anti-B antibodies were actually attacking my my daughter's type B blood cells, and so they were born with jaundice. Okay, first one was jaundice, just jaundice, and then she was okay. Give them her the Billy Light. The second one was severe jaundice, because in addition to the mismatch, she didn't want to come out, right? So they had to vacuum her out, right? There's that little vacuum that you can use to suck on their head and pull them out. For those of you who didn't know, now you know how we get out babies if they don't want to come out. So by sucking them out with a vacuum, that there's a big bruise on their head, and that elevated the bilirubin. So she was very jaundiced as well. Okay, yeah. So that's basically hemolytic disease of the newborn. Again, severity ranges from mild to very severe. If it's very severe, you would have to, do blood transfusion while the baby is still in the mother sometimes to save them. So that's that. Does that answer your question? That's the short version. Okay, short version took like 15 minutes. Make sure you watch the whole thing afterwards. Okay, there's another question. Bone marrow transplantation can be used to treat leukemia. Speculate the effects of bone marrow transplantation on a person's blood type. Well, all our blood, right? All our blood cells, red, white, platelets, right? All of them came from bone marrow, okay? Through the process of hematopoiesis, right? Okay, so this happens in the bone marrow, and it gives rise to all our bone or all our blood cells. Okay, so in the bone marrow, we have these stem cells that can become, different blood cells. So what happens is, if someone has leukemia in a very simple term is uncontrolled production of blood cells. Okay, so by transplanting healthy bone marrow, right, you introduce new stem cells that don't have that the problem of dividing uncontrollably. Okay, then you kind of fix the problem, kind of treat the leukemia, right? But depending on who you're receiving the bone marrow from, you might get stem cells that are of a different blood type than yours. Okay, let's say you're a type B blood and you received bone marrow transplantation from a type O person, right? Then now your blood type can change, right? Over time, it could change to type O. Okay, not always. It could just be like a transient thing. But if majority of the bone marrow has been transplanted that way, then it's possible for you to switch the blood type. Okay, so that's the answer. Okay, let's do number 2 while we're at it. Sally's first child was born with hemolytic disease of newborn, which is what I just explained to you earlier. She learned about RhoGAM from her friend. Sally is now pregnant with her second child, and she's requesting to receive RhoGAM shot from her doctor. Is her request valid? First of all, can someone tell me what RhoGAM is? What is this thing? RhoGAM. Anybody?
Are those the artificial antibodies?
Mhm. That's right. These are artificial antibodies. Artificial anti-RH antibodies. Okay. So who do we normally give RhoGAM to? We give RhoGAM to RH negative mother to prevent sensitization. Okay. So remember that mother's bloodstream. Okay. We're gonna put some RhoGAM in her. Okay, RhoGAM. Let's just say they're red shape, red colored. Okay, RhoGAM antibodies. So when the placenta is damaged and the baby's positive cells goes into the mother, okay, normally it would trigger her immune system, right? However, if you receive a shot of RhoGAM, these antibodies, okay, listen carefully, these antibodies will remove all the positive cells before her immune system kicks in. Okay, so RhoGAM removes all RH positive RBC before the immune system is activated. Okay, so that's how you prevent sensitization. Now, in this question, Sally, first child, was already born with hemolytic disease of the newborn, which means she already have anti-RH antibodies in her system. Okay, so giving her RhoGAM would not undo that process. Okay, RhoGAM is to prevent sensitization, not reverse it, right? Okay, so for that reason, there's no need. Well, receiving the RhoGAM would not help in the second pregnancy. Okay. So I have a question for you guys. If it was her first child, why would the kid have hemolytic disease of the newborn? I thought you only you can only get sensitized through the first pregnancy. If this is already the first pregnancy, why would she even have the antibodies in the first place? Anybody? Yeah, could be a blood transfusion, right? Maybe sometime earlier in her life, she received a positive transfusion for whatever reason, and that could have sensitized her, right? And now she has the antibodies in her, right? And this is something that you can check, right? If you do like prenatal screening, before you get, like when you're pregnant, you get your blood tested and stuff like that. They could check for all these things, okay? So it would have been easily picked up. Okay, that is it. Yeah, I will post the answers for the fill in the blanks as well, and that's it for this week. All right. If you still have questions about the blood type stuff, if you don't get it, send me an email. We'll set up something, and we can talk more about it. Okay. Good luck with the quiz, and thanks for coming. I'll see you all next week. Bye for now.